Hi, I'm Beth Moorefield, an editor at Nature, Structural, and Molecular Biology, and I'm speaking with Marisa Bartolomei, who works at the University of Pennsylvania Perelman School of Medicine and studies a unique process of gene expression in mammalian cells known as genomic imprinting, which directs expression specifically from either maternal or paternal alleles. I thought we could speak some about the function of the imprinted genes and the mechanisms that govern their regulation.
Sounds good.
Okay.
So let's just start with what are imprinted genes, how many are there. So imprinting, as we'll discuss it today, is a mammalian phenomenon, and it affects about 100 to 200 genes and is nicely conserved among mammals, which gives us the opportunity to use mouse as a good model to maybe study imprinting in humans. And these genes have very important processes in growth, but they also have functions in postnatal energy homeostasis, in behavior, and in other processes. And so that when these genes are missing or defective, you end up with very broad changes and broad sort of imprinting disorders if there are defects in humans. And that would include Beckwith-Wiedemann or Silver-Russell syndrome. Those are growth imprinting disorders. Or Angelman and Prader-Willi syndrome, which involves imprinted genes, and those are neurobehavioral disorders. So they have a broad range of functions. And as I said, absence of these genes causes these disorders, and that's why we want to really understand their regulation.
And are the maternal or paternal alleles imprinted with equal frequency, or is there a bias toward one or the other?
So these imprinted genes are regulated by regions that we call imprinting control regions. And these imprinting control regions are discrete elements in the genome that experience epigenetic modifications. So they have DNA methylation that's put on either during male gametogenesis or during female gametogenesis. And if you look at these regions, there's many more of these methylated imprinting control regions that are methylated through the female germline, just a few in the male germline. So a lot of the action seems to be happening in the female germline. That said, these imprinted genes are found in large clusters through the genome, and they're expressed either from the maternal or the paternal alleles, and there seems to be pretty well equally represented maternally expressed imprinted genes and a paternally expressed imprinted gene. So there doesn't seem to be a bias in that sense.
And how are they distinguished, either of these alleles, or how are they identified as maternal or paternal, so that they're differentially recognized by, say, the transcriptional machinery?
So that's the question. So we know that that these differential epigenetic modifications that are put on in the germline is what helps to say if this comes from the maternal allele, so either express or repress it off the maternal allele, or modifications that are put on in the paternal germline help to recognize something as being paternal and being either expressed or repressed. And so these germline modifications are actually key to allowing the somatic cells to say maternal express, paternal repress, or vice versa. So these modifications that are put on in the germline are really the key to the imprinting. And when something is perturbed, that's when you see misregulation of imprinted genes. And that can occur in the germline, or it also can occur at post-fertilization.
And do each of the alleles share common regulatory elements themselves?
When imprinted genes were first identified about 25 years ago, the first imprinted genes were identified, and they actually were 3 imprinted genes that were published in the same year. IGF-2, insulin-like growth factor 2, the insulin-like growth factor 2 receptor, and H19, which encodes a non-coding RNA. It was thought that these imprinted genes were going to contain this sequence that was this primary sequence that was identified either in the male or the female germline, and that was what was going to be the key to imprinting. And as it turns out, and there was going to, and in fact, what we thought there was going to be a silver bullet. You know, we're going to identify the imprinting box, like the Tata box. There was an imprinting box, and it was going to be a simple sequence. And there's no such simple sequence. And so maybe there's something more complex that we just haven't figured out with secondary or some other kind of sequence. But each imprinted region, each imprinted gene seems to have a different type of control sequence that does different things. And we really don't know what that specific signal is that says this is going to be an imprinted gene and this is not going to be an imprinted gene. We have little bits and pieces of it, but the absolute mechanism hasn't been worked out completely.
Then are they actually affected areas, the ones that are differentially modified, are they always in the same position within these alleles or within the different genes themselves?
So that gets a little bit to the mechanism. So that the real central piece of imprinting is, as I mentioned before, these imprinting control regions, or ICRs. And those are the ones that have the methylation put on either in the male or female germline, and it's maintained as long as imprinting is maintained. And there's two regions that people see that these ICRs can be found. And one is intergenic, so in between genes, and the other is actually in promoters. And often it's in the promoters of long non-coding RNAs. And these were some of the first uses that were found for long non-coding RNAs, that a transcription of these long non-coding RNAs was what was imprinting genes in cis. And so the methylation is in the promoter, and if it's highly methylated, the long non-coding RNA is not made, and if it's unmethylated, it is made. And so that's an example of how these imprinting control regions are not only giving you maternal or paternal-like differentiation, but also functioning to do something. So an example there then is serving as a promoter. For the intergenic imprinting control regions, that's a little different. The one that I study, H19 and IGF2, that imprinting control region is actually an insulator region, and it binds CTCF. So when it's unmethylated, CTCF binds, and it helps to confer the imprinting of the region. When it's methylated, CTCF can't bind, and so there's no insulator form. So these ICRs have these two sort of broad functions. There are other mechanisms involved, but those are the two main ones where the methylation is, where the ICRs are, what they're doing.
So if you have this differential pattern, how does it actually get propagated to the daughter cells upon division?
So when the methylation is put on in the germline, it's through the de novo DNA methyltransferases, likely chromatin modifications, differential chromatin modifications are involved too, but the propagation is through the maintenance methyltransferase DNMT1, and that's been shown. It was actually one of the first experiments done by the Yanish lab showing that in the absence of DNMT1, there was a failure to maintain imprinting. So as the cells divide, DNMT1 migrates with the replication fork and puts the methylation on the newly replicated strand so that the imprints are maintained. And the key for imprinting is that it's epigenetically maintained. It's maintained throughout the life of the organism in most cases.
And one aspect, well, not only maintenance, but of course the modifications then have to be reversed to permit germline development. Right. So how is that accomplished then? So
the mammalian embryo is very interesting in that there's two major times in development when there's this very large scale reprogramming that occurs. The first time is after fertilization. I guess every development is circular, so you can say it's the first time, you can say it's the second time. But let's just say you start with right after fertilization, the gametes come in with their own methylation patterns, and there's reprogramming as these cells are going to become pluripotent. At that time, imprints are maintained, okay? And so there's something very special about them that enables that differential methylation to be maintained. So with mammals then, as the embryo developed, the germline is set aside from the somatic cells. So at early post-implantation, some cells that are recruited from the somatic cells based on their location are going to go on to form the germline. And what happens there is then there is a second time of reprogramming. And in that case, everything that will be reprogrammed is reprogrammed, including imprinting control regions. And that's because if you're taking the germline from somatic cells that have maternal imprints and paternal imprints, and you're going to become a germ cell, let's say in a female, you want to erase the maternal and paternal imprints and put on the maternal imprints. And if you're in a male, you erase those and put on paternal imprints. And so that reprogramming occurs as the primordial germ cells are being specified and are replicating and migrating to enter the genital ridge. And it was originally thought that what would happen, because a lot of methylation is lost at that time, is that the cells were replicating, DNMT1 is downregulated, moved outside the nucleus for the most part, and so the cells would replicate and there wouldn't be maintenance methylation, right? So you would have just what we call a passive loss of methylation. Things would be diluted out, they enter the genital ridge and they're remethylated. But we know now that sometimes the demethylation is faster. Mm-hmm. And so in the last 10 years, a whole new mechanism of demethylation has been described through the TET enzymes, and it's a family of 3 enzymes that will oxidize methylcytosines, TET1, 2, and 3, and TET1 and 2 are expressed at the time when the methylation imprints are being erased. And what we and others have shown now is that in the absence of TET1, some germ cells fail to erase all of the imprints, all of DNA methylation imprints. Others do it fine. So it seems that active and passive methylation are working together to reprogram, to erase imprints. And what we see is in some germ cells, some pools of germ cells, we see a lot of remaining methylation. Some we see hardly any. And we don't really understand what's going on in that case. It's really, the more you dig, the more mysteries you find in this field. It's really an incredibly exciting field, a lot to be learned. Mm-hmm. So that's still an ongoing research of what we're trying to understand. And why is it? Why is it do we need 2 mechanisms, an active and a passive? Are they redundant? Are they helping each other? Are there some regions of the genome that are just so densely methylated, have such high affinity for DNMTs that you need this extra mechanism in place to demethylate? We don't know yet, and we'd like to really learn that. We and others are really like to learn that.
Oh, that's excellent. So it's not clear yet whether or not actually the residual methylation is actually precluding the de novo methylation events, or like what the functional interactions might be there.
Again, because we can't really follow a given cell, we do know that if we look at TET mutants that come from the female, and we look at maternally methylated regions that we expect to be methylated in oocytes, they're methylated fine. But if we look at paternally methylated regions that should be unmethylated in oocytes, they are not. Sometimes they're still methylated, sometimes they're not. So it doesn't look like that residual methylation, that whatever might be happening, the reprogramming is preventing de novo methylation from occurring. But that's only looking at early in development and later in development. We don't know what's happening in between. So if we could live image cells, and there's always this on paper experiment you could think about where you could follow a cell and see demethylation occurring at every different look, but we can't do anything like that yet.
Okay,
so.
Not yet.
Not yet.
Also, how about other things that not yet, as far as I'm aware, but is there any evidence that environmental factors can influence any of these activities and actually alter its patterns?
So that's another area of active investigation in my lab and lots of other labs. And so there's a whole field called Developmental Origins of Health and Disease. And the idea there is that insults in utero to an embryo can be maintained or remembered and expressed later on in development. And we're very interested in this hypothesis. A lot of it was shown, was originally defined by David Barker, and it was shown epidemiologically during the Dutch hunger, when there was minimal caloric availability to people at this time. Women who had first trimester babies during these times of minimal calories, these children were born and 50 years later had very high levels of heart disease and metabolic diseases. And so a lot of that was epidemiologically shown, but it's now been moved to animal models. And we've been very interested in this idea that very early gestational exposures may be the most dramatic, and that's because that's when we and others have shown there's a lot of reprogramming that's going on. And so we've looked at endocrine disruptors and some assisted reproductive technologies where there's exposures in the early embryos. And we use imprinted genes because that for us, they're our canary in the coal mine. We know that they have some very special replication in the early embryo. And we've shown with some of these exposures, and other people have shown with some of these exposures, there are defects in the ability to maintain the methylation imprints or maintain other kinds of methylation and there are defects in other kinds of reprogramming. So environmental perturbations can, during that early time in development, can have a lasting influence on offspring, and we're trying to look at that in more detail. So it's another area of very, very active investigation as people are have the capacity now to profile what's happening in the early embryo. Before we couldn't do that. Now we can take small numbers of cells, look at DNA methylation, look at chromatin modifications, compare normal to environmental exposure of some kind.
That's excellent. Really exciting time for this. Well, thanks.
It is an exciting time. There's a lot, science is such that we can do a lot of things that we hoped we could do 20 years ago, and now we're doing it. So
we're so well placed.
Yes.
Okay. Thanks again.
Thank you. Thank you.